Hello, my name is Louis Schwartz. I am a chemistry major as well as a theater minor. Firstly, I want to thank you for stopping by to check out my IS research. My research was on drug delivery, specifically the release kinetics of ibuprofen from swellable organically modified silica by means of a non-active release modulator, and I was advised by Dr. Paul L. Edmiston. To start off, I'm just going to give a little overview of drug delivery. Drug delivery helps with the treatment of certain diseases and illnesses, mostly used with over-the-counter drugs, but also with prescription drugs as well. The rate of release is important for drugs because these drugs are being delivered to the bloodstream. So the release rate has to be a therapeutic dose without the possibility of an overdose. A couple of common applications of this include capsules, which you can see on the top. Right below that is a patch. Most commonly a nicotine patch is what you would see this in. And then as well as microneedles, such as insulin syringes, insulin pumps. On the side there you have a picture of an insulin pump and what it looks like below the epidermis, or the first layer of the skin. And now I want to get into a few goals that I had for this research. My first goal was to develop a method to encapsulate ibuprofen and a modulator into a porous silica bead. I also wanted to measure the release rate of free ibuprofen into a flowing stream of water, which ties into my next goal of measuring the release rate of ibuprofen encapsulated in this silica bead material with a modulator. And then I wanted to compare the two to see if there was any difference of the free release rate of ibuprofen into a flowing stream of water versus the release rate of ibuprofen from this silica bead with the modulator. So both of those tie in together a little bit. And now this is my method that I used for measuring ibuprofen release over time. My trials that I ran either ran for 24 hours or over the span of an entire week. So water would go into the top of a glass column. Inside this glass column would either be free ibuprofen or encapsulated ibuprofen inside the silica beads that I mentioned in the previous slide. And then through that, water would come out through these silica beads, releasing a concentration of ibuprofen at every hour. For the 24-hour time span, I would collect samples at every hour, and for the week-long time span, I collected samples at every 4 hours to be analyzed. So here's my results from a week-long release of free ibuprofen inside the glass column. The solubility of ibuprofen is about 0.021 milligrams per milliliter, which translates to about 2.0 micrograms per milliliter, and this is what we would expect the release rate of ibuprofen to be when it is free. As we can see from this graph, that is not the case for most of these points. In the first 4 hours, it seems it's releasing at a little bit of a higher rate, but as time goes on, that release rate significantly decreases. So the silica bead that I've been talking about is known as swellable organically modified silica, or SOMS for short. How these work is with an active ingredient or solution, an organic solution. You add this to the SOMS, they swell, and as that happens, the organic solvent evaporates out, the SOMS contract, and it traps whatever is left inside. So my solutions were with ibuprofen and whatever modulator I used. I had a variety of 3, and I used different loading volumes of each. So at the end of the evaporation, what was left inside the SOMS to be tracked for release was the ibuprofen along with the modulator that was used. And here we have the swelling mechanism of SOMS, seen as a visual representation. So the picture on the top is what happens when you add the organic liquid to the SOMS over a period of 10 seconds. As you can see, the swelling happens very fast. The picture in the middle is a molecular level diagram of what SOMS look like when they're dry, partially swollen, and when they're fully swollen. And then on the bottom you have another figure representation, which also was seen in the previous slide, so I won't go into detail about that. So here we have a visual representation of what happens when you encapsulate modulators along with another material inside of SOMS. As we saw 2 slides ago, The figure on the far left shows a fully swollen SOMS with ibuprofen and a modulator entrapped in it. The ibuprofen is denoted in blue, while the modulator is denoted in red. So as the organic solvent evaporates out, the ibuprofen and the modulator sticks around, and the ibuprofen is entrapped while the modulator holds the pores of the SOMS open, as seen on the far right, and allows the ibuprofen to be released without any hindrance. Again, here is the same graph that we saw 4 slides back. However, now it includes a curve that includes the release rate of ibuprofen when it is encapsulated with a modulator. So as you can see with this modulator, although it's not at 2 exactly, it is almost at that 2.0 micrograms per milliliter mark and is releasing pretty constantly for about the first 24-ish hours until it dips off to about the level of the free ibuprofen rate. So the release rate is a little bit more steady at the beginning, however, towards the end it does tend to die out like the free ibuprofen release that we saw. So here are some interesting findings from the research. I'm not going to go into depth about this whole table, but we'll focus your attention to the farthest left column and the farthest 2 right columns. The farthest left column denotes the modulator that was used and was entrapped along with ibuprofen in SOMS. PEG 8000, PEG 200, and glycerol were these 3 modulators. PEG stands for polyethylene glycol, and the numbers associated with them are their average molecular weights in grams per mole. And glycerol's average molecular weight is about 92 grams per mole. So as you can see in the farthest right columns, as we increase the molecular weight of the modulator, the percentage of ibuprofen released from SOMS does go up significantly. As we see from the farthest right column, glycerol in a week only released 27.9% of ibuprofen, PEG 200 released about 46.1%, and PEG 8000 came in at 98.1%, so almost 100% of the ibuprofen was released in this case. The main conclusion of this research that we saw was that the larger the molecular weight of the modulator, the faster the release rate of ibuprofen from these SOMS beads, and the more ibuprofen was released from these SOMS beads. Some future work that can be done would be to analyze the drug-releasing SOMS beads before and after the release data of ibuprofen is collected in order to determine if any of the modulator also remains entrapped in the SOMS beads. Something else that we can do is to increase the molecular weight of the polymer modulator with the goal of achieving 100% ibuprofen release at a constant dosage, which, as we saw 2 slides before, a constant dosage was achieved for about 24 hours, but after that it started to dip off. Again, thank you for coming by to see what my IS research was. A couple acknowledgments I want to make. Of course, my advisor, Dr. Edmiston, without whom this research would not be possible. I'd like to thank my advisors throughout the years, Dr. Lee McBride, Dr. Sarah Sobek, and Dr. Paul Bonvalet, for helping me find my path at Wooster. I'd like to thank the whole Department of Chemistry for their support throughout this project and the funding to actually do this project. Again, thank you for coming. I encourage you to go check out all the other presentations that are going on today, and have a great day.